I mean,
And I guess Bob Dirksen was at Rochester, right?
Yep.
And okay, and he's got kind of two careers. He works on muscle,
That you
and
know of.
yeah,
Let's say.
oh yeah. Maybe the other ones I don't want to know about. Um, uh, Myotonic dystrophies and gene therapy for that. And then what you're going to hear about today, I was very excited to hear about this and learn of John's work on engineering transfer RNAs to recognize stop codons. And there are a lot of diseases, I'm sure he's going to go into this also, that are produced by nonsense codons. And I really think this approach is going to cure cystic fibrosis, variants of cystic fibrosis, muscular dystrophy, Marfan syndrome, many cancers and developmental deficits are produced by nonsense mutations. So to find a way to read through stop codon, which he's going to go into in much more detail, I think is a really exciting process and possibility. And John did what is a kind of heroic screen when he was in Iowa, where they checked every single tRNA for each amino acid against all the three stop codons and see which one of these would work better. That's like really heroic kind of thing to do. And it was quite successful. And you'll see they get some really amazing read through. So that's why I was kind of, we work a little bit in this area, not quite the same thing, but that's how I came upon John's work. So it really is great to have him here. I hope you all enjoyed talking to him. And I'll give the floor to him. So
Yeah, thanks. Yeah,
thanks for coming.
Yeah, today's been great. I appreciate the invitation to come here and see, make new friends and meet old friends, not friends that are old, but just old friends. So yeah, so I'll get right into it. And so I'll be talking about the development of a platform, anti-codon edited, we call ACE tRNA technologies for nonsense-associated diseases. And quickly, this works. Here are my disclosures, which is not really important. Okay, so go up here. I'm going to try to get rid of that. No? Is it hard to see? It doesn't matter. We're going to leave that there. Okay. So what is the problem? So the general problem that we're trying to solve are nonsense mutations. And so to start from the very beginning, in normal translation, you have the ribosome, which I've indicated here in a blue and purple ball, bound to an mRNA, which is a squiggly black line, right, is translating a protein, which is shown here in high resolution as a green cloud, right? And so in every transcript, you have a normal stop codon at the end, which terminates translation, dissociates the ribosome. And premature NPTCs or nonsense mutations, you have a stop codon that occurs from a single base change in the middle of the reading frame. So when the ribosome comes through, it sees that stop codon first. The ribosome dissociates. You have a truncated protein that's not functional, and in some cases actually has a dominant negative effect where the truncated protein can act on existing proteins and even cause even more severe forms of the disease. And then you also have the second effect where because the mRNA was not translated all the way through, it goes through this process called nonsense-mediated decay. So it's kind of like a two-hit mechanism. But the end result is that you do not have a functional full-length protein. So the problem is nonsense mutations. This occurs in 10 to 15% of all genetic diseases. And it's caused by these nonsense mutations that, again, premature termination codons that affect 3 million people in the United States and about 300 million people worldwide. They arise from the sense codon having a single nucleotide change to one of the three stop codons, TAG, TGA, and TAA. So nonsense mutations or the nonsense-associated diseases are the more severe forms of any of the diseases that you know of. It's because you don't have like a 50% or a 25% loss in protein function, it's 100%. And so the ramifications are much worse. So some of the ones that you might be familiar with are Hurler syndrome, beta thalassemia, Usher syndrome, spinal muscular dystrophy. What we're very interested in and applying these technologies to that I'll be talking to you about is Duchenne muscular dystrophy, retinopathies, and cystic fibrosis. And so there's a seemingly high prevalence of nonsense mutations that result in this disease, and it's all due to the same mechanism. So the idea is rather than coming up for therapies for each one of these individual diseases, is to come up with a therapy that is a platform therapeutic that is disease agnostic and mutation agnostic. And so while each one of these diseases is considered rare, right? All together, they're very common, and that's the idea here. And so there's lots of approaches that are being taken as therapeutics for nonsense diseases. A lot of our small molecules will actually bind to the ribosome and read through. We're taking a tRNA approach, and I'll go through that now. So in normal translation, you have a tRNA that's recognized by its canonical amino acyl tRNA synthetase. That tRNA is charged with its amino acid. That acylated tRNA then is bound by EF1 alpha, which is that red blob there. It acts like a little helmet for the acylated tRNA, keeps it from being deacylated. It also brings it to the ribosome where it participates in translation. In normal cells, there are no tRNAs that recognize any of the 3 stop codons. So when the ribosome comes to a stop, the release factor comes in, binds, dissociates the ribosome, and you have termination of translation. What we're doing is we're taking human tRNAs, we're mutating the anticodon now to recognize one of the 3 stop codons. It will then still be recognized by the canonical amino acyl tRNA synthetase, charged with the correct amino acid, bound by EF1 alpha. And when the ribosome comes across a stop codon, now you have tRNAs there present to recognize that stop codon. You'll get read-through and full-length protein. And the really important thing to understand here is that with all of this that's going on, the only thing that we need to deliver to the cell is the tRNA, right? And so it hijacks the rest of the endogenous system. The other really important thing to realize here is that you need to be able to get enough of these ACE tRNAs in the cell to outcompete the release factor. And so that's one of the biggest hangups here. And that by competition, it's not competition in the truest sense. It's that whatever gets there first wins. And so you need to have a high effective concentration of these ACE tRNAs. The other thing I wanted to mention at this time is that we didn't come up with this idea, right? This idea has been around for a really long time, and people have been using it for all sorts of different kind of physiology, biology, cell biology, generation of tools. And what we're really trying to do is take this and make it a therapeutic to hopefully help people. So the idea isn't ours. We're just trying to get it across the therapeutic goal line. So tRNAs are a very cool molecule. So tRNA genes are very tiny. So the tRNA itself is its own promoter. So in order to express a tRNA, you only need about 150 base pairs of information. There's the upstream control element, which I have in dashed lines, A and B box, which are really important for binding the transcription factor, and a poly T track, which is for transcription termination. You can deliver ACE tRNAs in several formats. You can deliver them as RNA, right? So you can just do runoffs, fold them, and deliver them in lipid nanoparticle formulations. And this is effective. We've done this in the lab, and there's entire companies that are working on this approach. You can deliver them as DNA, where you can encode them in little plasmids, and also you can deliver them in viruses. And so one of the limitations of doing gene complementation is that if your cDNA with the promoter and transcription termination is bigger than 4.5 kb, you'll never get it into an AAV, right? So for Duchenne muscular dystrophy, they went through a lot of tricks to truncate or make smaller the dystrophin in order for it to fit. No matter what gene target has a stop mutation, we will always be delivering 250 base pairs of tRNA expression cassette. So in this way, the cargo is disease agnostic, right? We're always delivering the same thing no matter what the target is. So it leaves open all these opportunities for therapeutic moieties. tRNAs are, when we first had this idea, I knew nothing, so this is for the graduate students out there, knew nothing about tRNAs in translation biology. This is literally an idea that I had that spawned from this group, or this field that I was working in called genetic code expansion. And I was, in full disclosure, I was taking a shower. I was like, well, what if we just mutate human tRNAs to recognize these stop codons, and then we can use them as a therapeutic? And then I went back to the lab and realized that people had been doing this forever. And I also realized that it's just not as easy as that. It's because there are 415 tRNA genes in the human genome. And which one do you pick to move forward with? Right? And then we started looking through all the phylogeny, you can see that different organisms have different amounts of tRNA genes. And what was super interesting to me is that bush babies have over 45,000 tRNA genes in their genome. So bush babies are these animals over here to the right. And the question is like, why do bush babies have 45,000 tRNA genes? And then I think, they're actually probably doing lots of different things. They're acting as insulators, opening chromatin. You can activate them under different stress conditions. I always say that they are really important for the expression of cute genes, as you clearly see in the bush babies over here on the right. But tRNA genes, their regulation, what they do functionally are super interesting. So rather than just picking one, which is what the field had done up till now, as Yale mentioned, we chose to look at all of them. So we took all the unique human tRNA sequences that with the anticodon was one nucleotide away from the stop. And we set up a screen where we had a nanoluciferase, which is a deep sea shrimp luciferase that would produce light. We put a stop codon in it. And so if you suppress the stop codon, you would get light. The more light you would get mean that you would have more stop codon suppression. So on x-axis is the amount of light that was emitted from a rescued nanoluciferase when we co-expressed one of these ACE tRNAs. And so this is in log scale, and the red line is 0. And so just to kind of show on like an example, if you look at the top left with glycine, a lot of the sequences actually didn't give us a very functional suppressor tRNA. And in fact, only 3 did. And if you looked at any of the literature, there was no way to predict which one of those sequences was going to support an active ACE tRNA. And so that's why you have to do the experiment, right? You can think about it all you want, but when it comes down to it, a lot of times is putting pipette to liquid and doing the experiment. And so the other thing to point out is while it's heterogeneous, that you can actually get really good PTC suppression. And so you can see that the scale here from 10, 100 to 1,000, you get 1,000-fold over background with some of these sequences. The other thing to point out is that if you look at my favorite ones, which are leucine and serine, you can convert those to suppress any one of the 3 stop codons, right? So you can, with the same body sequence, you can mutate it to suppress a TAG, a TAA, and a TAA, TAG, TGA. Yep. Okay. So what have we done? So we did the high throughput screen to identify the most active ACE tRNA. Using ribosomal profiling, we and others determined that ACE tRNA suppress natural termination codons, the codons that are at the end of every open reading frame, with lower efficiency than those that are in the middle of the open reading frame. And so this is the question that I always get, and after this we can discuss this because this is what I get at the end of my talks always. But NTCs, natural termination codons, have been evolved to be better stop codons. Using mass spec, we found that when we make these modifications to the sequence, that we're actually still charging them with the correct amino acid. So by changing the anticodon, suppress the stop, a leucine tRNA still is charged with a leucine. And so in that stop codon, you'll get a leucine co-translationally inserted. We showed that ACE tRNAs effectively suppress PTCs and rescued wild-type levels of transcript abundance. So it actually will inhibit nonsense-mediated decay, and also will give you almost wild-type levels of your target protein. And we recently showed that we can take our original screen, and we did a larger screen on individual sequences and could optimize them by nucleotide substitution to have over 20-fold better PTC suppression efficiency. And this is important because if you're going to go in for therapeutic delivery is very hard. And we were trying to lower that delivery barrier by making each one of the ACE tRNAs more effective at suppressing the stop codon. So we did this screen thinking, oh, we can just make wild-type proteins now. So in cystic fibrosis, the most common nonsense mutation in CFTR is G542X. And so we were thinking, okay, we're going to use our really active glycine ACE tRNA and make a wild type. Or we can go in and use one tRNA and see if we can suppress a lot of the most common nonsense mutations and see if we'll get fully functioning protein. And so instead of going a wild type, we would have a G542L. And then the second most common, right, W1282, we wanted to put a tryptophan in. Or could we also put a leucine in that position and get fully functional channels? And why is this important? Is that if we're actually going to be making a therapeutic of this, it would be almost impossible to advance 20 different ACE tRNAs to the clinic, right? It's just too expensive. There's just no way that that's going to happen. If we can advance one, then we would, you know, that was obviously more likely. And so having one therapeutic relieve the burden of bringing several sequences forward to the clinic where there's not enough money probably in the world that would be wanting to be spent on this to do it otherwise. Can we make ACE tRNAs more efficient at PTC suppression? So by lowering the delivery burden by requiring fewer ACE tRNAs to deliver to each target cell? And can we effectively deliver ACE tRNAs? So that's the big question at the end. And while this talk is focused on nonsense mutations that result in CF, these ideas are clearly transferable to other genes. And again, I mentioned we're very focused on Duchenne and retinopathies. So to get into the research, so looking at cystic fibrosis, they have this cell line, human bronchoepithelial cell line, in which they've CRISPR'd in nonsense mutations. And so these are really good because you can look at the transcript abundance using quantitative RT-PCR, which is shown up on the left. You can look at protein abundance by Western blots, but most importantly, you can look at the function of the CFTR channel, right? So function is really what matters here. So on the top left, using Using chamber measurements of short circuit CFTR current, you can see wild type on the left when you've activated CFTR with forskolin and IBMX. If you wait a bit, you can see that there's a second hump. And that's actually a FDA approved modulator for CFTR that binds to the channel and increases its open probability. And this is a drug that Vertex came up with, which is part of this drug called Trikafta. You see you get a bigger hump. With the nonsense mutation, you see you do have any current at all. There's these molecules, these aminoglycosides that bind to the ribosome that cause read-through. And when they use that, you can see on the right, you get 1.6% of wild type. Then you can use a nonsense mediated decay inhibitor called SMUG1. And when they use that alone to increase the abundance of the transcript, you get about 1% of wild type CFTR. When you use those in combination, you get 2.7% of wild type CFTR function. And so that was hailed as a big success. That tells you how hard it is to read through these nonsense mutations. The caveat with these experiments is that G418 is toxic, and so is the SMUG1 inhibitor. So while you're able to rescue the CFTR function to an exciting 2.7%, you're also going to kill the cells and kill the patient. So keep that number in mind with 2.7%. Using ACE tRNAs, we did something similar. We put them on plasmids. We just transfected them into the same cell types, W1282X, and then another mutation, R1162X. And we did whole cell patch clamp, pre-activated the channel with forskolin and IBMX. We did ramp protocols, and we plotted the peak current from the ramp at plus 80 millivolts over time, right? So you can see in wild type, you get really nice channel activity. There's a specific blocker called inhibitor 172. You get a decrease in CFTR channel activity. When you use whole cell patch clamp on the two nonsense mutations, you see you get no current. When we transfected in our ACE tRNAs with the arginine, so we put in an arginine ACE tRNA in red, you can see that we got near wild type expression or a rescue. And then for the first time, we tried this idea of putting the incorrect amino acid in, where we made it, instead of making a W1282W, we made the W1282L. And you can see that we got near wild type rescue of channel function. So this was the first idea that we don't actually have to make a wild type channel to rescue its function. And that's all the average data is shown on the right. So then we moved forward and we picked the four most prevalent nonsense mutations in CF, which are shown there. And then we tried each one of our most active ACE tRNAs in each one of the sites to see which one would be the one that we would advance to move forward. Yeah.
Did you do the W position?
Yeah. And with W?
Yeah.
And it's terrible because we did. And the reason it's terrible is if you look at our tryptophan UGA suppressor, it's the worst in the lot. And so even though we would have wild type channels, the suppression efficiency is terrible. And so we would get something like 5% rescue. So it almost forced our hand to do this other experiment with leucine. there any other questions? Okay. Okay, so what we did is we plated our cells on trans wells. We then used adenovirus, which expresses our ACE tRNAs. We waited 4 days and we performed leucine chamber experiments, right? And so this is just a representative trace. And then we take area under the curve and we plot them here. So we have the scrambled control for wild type. And this is in the absence and in the presence of that drug that Vertex is now FDA approved. And so with the idea being that if we can rescue a little bit of our channel, we can augment the rescue by using these modulators. Okay, so we use scramble, ARG-GlyLu, ARG-GlyLu across all these mutations. And you can see in blue is when we put leucine into G542, R553, 1162, and W1282, that it rescued above 10%, about 15 to 30% of wild type. And so that is more than 2.7% that we got with 2 toxic small molecules. And we got really excited about this, but that's not 100%, right? Why did we get so excited about it? It's because that when we went back and performed flow cytometry on these cells, we had a GFP in the backbone. We actually only got it into about 25% of the cells. And so what this means is that we can actually get enough of these tRNAs into each one of the cells, we can have an effective therapeutic. And the real problem is delivery. And that's what I'll be talking about at the end of my talk. So we were excited about this, and knowing that people were really excited about 2.7%, we're excited about 25%, and we know that we can do better than that. We went into a different cell type, which is a patient-derived organoid. And with this assay, if you rescue CFTR function, they swell because CFTR passes chloride, water follows. And so you just actually look at the volume of these organoids. In the absence of CFTR, you get no swelling. And so it's a bit similar to an leucine chamber experiment. So in these cells, when you use G542 homozygous R1162 and W1282 with leucine, you see that you rescue CFTR function, and that's shown by swelling. And if you use a compound heterozygote patient with one nonsense mutation, so in only one of the allele, and then in the other allele you have a splice mutation, having one allele is enough of a target to rescue enough CFTR to recover swelling. Okay, so what is the function of each one of these CFTR variants? So we've shown that leucine works for 4 of the most common. None of them are leucine to leucine, right? So basically, we're making missense mutations. And we know that missense mutations also cause cystic fibrosis. And so in missense mutations in CFTR affect the single channel conductance, the number of channels that are being trafficked to the surface membrane, and the open probability. And so when we break down what we've done here, we wanted to understand how putting a leucine in affects some of these that result in macroscopic current. So one of the issues or one of the factors that affects the number of channels is just the PTC suppression efficiency. And there's a lot of literature out there that shows that the nucleotide context in which your PTC sits actually influences its ability to be suppressed. And this is actually, as I explained earlier, why natural termination codons are one of the reasons why natural termination codons are harder to suppress than PTCs. And so to see if that's happening with our 4 mutations, we set up a flow cytometry experiment with a dual fluorescence where we had M neon green in the beginning, the context of CFTR with the stop codon, and M cherry. We then transfected that with those 4 ACE tRNAs, and we would just basically see what percentage of cells went from green to green plus red. And that would say, okay, if it was 100%, then it was a really easy one to suppress. If it was lower than that, then the suppression efficiency was affected by the context and the context alone. And so we co-delivered low amount of the ACE tRNA, right, with our reporter. And what you can immediately see is that if you look at the axes on these radar plots, is this the percentage of cells that were rescued, that our R1162X was the easiest one to rescue, right? And that the context of R1162X makes that nonsense mutation easier to suppress, followed by R553, G542, and W1282. And we did this for each one of the ACE tRNAs. when you then increase the amount of ACE-tRNA, you can see that the rest start catching up, right? And then if you can deliver more of the ACE-tRNA, you can overcome this nucleotide context. And in the end, you can rescue almost 80% of any one of those nonsense mutations. But it's a dose, there's a dose response that comes along with that. The context of the PTC does matter. The nucleotide context of the PTC matters. So the next thing we wanted to know was, okay, so we put a leucine in to those positions. How does the channel behave? So outside of the suppression efficiency, is the channel you're making actually functional? So in order to do that, we just made the variants. We encoded a leucine codon into each one and all the other amino acids into the PTC. So we made G542L, G542R, R553, and all on the top. We performed leucine chamber experiments to see, taking in, again, PTC suppression efficiency out of the equation, how do these channels function? And the really important take home from this is that when you compare to the wild type channel, all the leucine variants, which are shown here, gave robust channel activity. So even though now you've made a missense mutation, it can rescue 50% or more of CFTR function. And this is augmented by the application of the already FDA-approved drug Trikafta, which is shown in hash bars here. So again, this could be like a combinatorial therapy for cystic fibrosis. And this process actually is called serotyping. So if you want to know if a drug works on many different mutations, or if you're inserting a different amino acid into different nonsense mutations, you just test it. So if a patient comes up and they have for any disease, and there's already an FDA-approved drug, but they're not approved for it, you test it in cells. If you get a response that's above the clinical threshold, then you can go to the insurance companies and say, look, this patient actually is responding to this drug or to this ACE-tRNA, and they should be brought on for this therapeutic. And that's exactly what the Cystic Fibrosis Foundation Therapeutics Lab did. There's missense mutations that are responsive to the Trikafta, but there are very rare variants that were never tested. And so they used this approach and added Trikafta, and they did 655 missense mutations. And they picked up about 10 to 15 patients. Now they're on Trikafta, which is a life-saving drug, because of this approach. And so we did the channel function here. We also wanted to look at protein abundance. And so what I didn't mention is that on the CFTR, we put on this nanoluciferase on the N-terminus, right? This is a cool trick if anybody wants to not do a Western blot ever again, is that you can take the protein, run it through an SDS page gel, nanoluciferase stays folded. You just mix the gel with pyrimidine, and you can do in-gel luminescence, and that's what's shown here. So no antibodies. And so CFTR has a C and B band. C band is glycosylated, and it's the functional form. And you can see with the different variants that you get different amounts of C band. And those correlate with how much function that you get. And when we added Trikafta, again, the drug, you can see that there was almost a complete shift from the B band to the C band. And while we knew this was an FDA-approved drug and it worked, it's really cool to see it in your own lab. So two of the drugs actually bind the channel, make it fold better, and move to the surface membrane more effectively. And the other one is ivacaftor, which is potentially to increase its open probability. So the fact that this company actually found 3 molecules that bind to one protein and work in completely different ways and are combinatorial is just amazing. And then the second most amazing thing, we've actually got it to work in our lab, and it's super effective, right? So, and the 2 variants that aren't effective or that didn't have a really drastic increase in C band are W1282G and W1282R. The really kind of fascinating thing I think about this is that W1282R is a known missense mutation that causes CF, right? And we made it with an ACE-tRNA suppression, and we were able to rescue enough to be therapeutically meaningful, at least in the clinic. So anything above that is going to be a super rescue therapeutically. So then the question is, why leucine? Why are we thinking about leucine rather than any of the other amino acids? Outside of leucine is the one is one of the 2 tRNAs that you can make suppress all 3 stop codons. The other is it's pretty innocuous as an amino acid. It's hydrophobic, so it'll behave well inside of a folded protein. It's not charged. It's not bulky. Doesn't perform pi-pi interactions. No disulfide bonds and no hydrogen bonds, and it's pretty flexible, right? And so you're not putting in a bulky amino acid to change the structure and function of a protein. So I like to say that leucine is kind of like the dad bod of amino acids, right? It's like not too big, not too small, and it just is just right the way I think of it. And the other amino acid that we really like is serine, and it's very similar to that, right? Except for it's hydrophilic. And so if you have nonsense mutations that occur on the outside of the protein, on the inside of the protein, you could use either one of those exchangeably and maybe come up with maybe 2 therapeutics that would help you move through. So then we were really interested in, we were surprised that leucine worked and serine worked. I didn't show you the results with serine in all of these positions, So then we went to what would be predicted by alpha missense, right? And so you have a benign to pathogenic sort, right? So going to the right is more pathogenic than benign. And we plotted each one of the variants that we actually did our recordings from. And so the function of those variants is on the y-axis. And so alpha missense was really good at predicting a group of those variants that we made, but not good at others. And so I think, again, this shows that you can't rely always on, what's prediction. You actually have to do the experiments. And so with this information, we're actually going to go through and make all 200 nonsense mutations in CFTR and see which ones will we make the variant substitution to leucine and serine, which ones will give you function and which ones will not. So the summary of this part of the talk is that we found that leucine can give a functional protein with the 4 most common CF mutations, and these make up 70% of individuals with nonsense-associated CF. And we're very excited about other nonsense-associated diseases, which I've listed here. So the next part is delivery. So we know that it's functional once we get it into the cells. Which one of these approaches are we going to take? And so nature has come up with a very good way to get DNA into cells, and that's viruses, right? And so purposefully or not purposefully, viruses get into cells really well, right? We just recently came out of a pandemic that really showed how effective they are. If you're using them as a therapeutic, they're great, right? There's already a couple FDA-approved AAV therapeutics, but the problem is that AAV seroprevalence varies geographically, right? But they're similar enough that we already have innate immunity toward, many of us already have innate immunity towards the capsids of these AAVs. So the population of any sort of therapeutic AAV that we would use, 30 to 60% of us already have innate immunity. And so we would not be eligible for getting these AAV drugs. The other aspect of this is redelivery. So we don't know how long AAVs are going to express. The fact is that we're probably going to have to redeliver. And when you do that, 100% of the people who received AAV will have immunity towards the capsid. And we've come up with several AAV and viral therapeutics, and we were going out to venture capital and saying, hey, look how effective this is. And this is data I'm not showing here. And this was a couple of years ago, and everybody was really cold on the idea. And we were kind of shocked by that. And recently we found out, oh, it's because a lot of these venture capitals and pharma companies are off of AAV. And in fact, a couple of weeks ago, Vertex discontinued AAV research. Takeda ended early stage AAV work in 2023. Pfizer pulled their only AAV for hemophilia B. And so AAV seems to be on the outs, and that I think very interesting to me. And I don't think any of us really know why that's the case. It could be that they've come up with really effective other means to get nucleic acids into cells, or just knowing that they're not going to be able to get to 100% of the patients is a factor in itself. So knowing this, we thought we were going to just go with the DNA route. And this is not a very popular route because of the other approach that people are taking, which is RNA. And the reason that DNA is not popular is that any of the cells that you're going to go into as a therapeutic are post-mitotic, right? When you everybody in the lab who's ever transfected a cell knows that your cells need to split in order for your DNA for your cells to turn green, for instance. And that's because when your cells split, the nuclear membrane breaks down, right? And then when it reforms, the DNA is inside the nucleus, and that's where it transcribes and it expresses. And so any dividing cell is really easy to get DNA into the nucleus. Post-mitotic cells, you end up getting DNA into the cytoplasm. It just sits there, it doesn't get into where all the action happens. And so quoting Joe Zemner, one of the most important barriers to transfection, therapeutic transfection, is the movement of the DNA from the cytoplasm into the nucleus. And this has been a very hard problem to solve, and this is where most DNA therapeutics have failed in the past. So to get around that, what people have done is made very small DNAs, hoping that they can get into the nucleus, or they use active transport. So they put on DNA targeting sequences that bind transcription factors that are in the cytoplasm, and they get dragged into the nucleus, or they put nuclear localization signals on the DNA. And when you do that, people have shown that you can get DNA into the nucleus of post-mitotic cells. DNA is huge. So when you think about DNA as just of the mass, each DNA base pair is 650 Daltons. So the average molecular mass of a double-stranded codon is about 2 kilodaltons, right? Where the average mass of one amino acid in which it encodes is 110 Daltons. So there's a huge downsize of material from the DNA to protein. So when you think about expressing CFTR, if you think about just the coding region of CFTR, it's 4 megadaltons of material. And when you then put on a promoter and a transcription termination sequence, you're upwards over 6 megadaltons of DNA. So that's like, a giant molecule that you're trying to get into a nucleus. tRNAs are really tiny, 200 base pairs. So in any sort of puck plasmid, it makes up only 7% of the material. So the idea is, can we just make a DNA vector that has this bit of information? And we know that plasmid DNA has low intracellular mobility. It gets silenced because it has bacterial sequences in the backbone, and this results in toxicity. So I had no idea, I don't know, in like 2002, when I was doing non-stationary noise analysis as a graduate student, that I would end up being the person making grams of therapeutic DNA vectors. That's like a very far distance to move. But that's where we are now. All right. So what we did is we made several platforms of mini DNAs. So we made mini circular DNA, and so we just PCR'd 400 base pairs of the transgene, and then we ligated it to circularize it. The issue is, if anyone here has ever broken a stick on their knee, if it's a very big stick, it's easy to break, right? If you take a little, like everyone maybe did this as a kid, and you're like, picked up a little stick and you did it and it really hurt your knee. It's because little sticks don't bend, right? Big sticks do. And DNA is no different than a stick. And so if you try to ligate small pieces of DNA to circularize them, the end of each one of the DNAs is easier to ligate to its neighbor than it is to itself. And what you end up getting is concatenamers. And we're trying to get singular circular DNAs. And so we use this trick where we took a DNA bending protein called TFAM, and that's located in the mitochondria. It's important it's like the histone for mitochondrial genome. And it binds to it's agnostic to the sequence. We put it in with the ligase mix, it bound the DNA, it bent the DNA ends to be close to each other, and it allowed us to ligate to make these mini circles. And you can see that these mini circles are resistant to exonuclease unless we cut them with a restriction enzyme BSC361, and you can see that it goes away. So we can make circles that are resistant to exonuclease, and we can do this a different way where we put them into bacteria. This is a technology developed by Mark Kay at Stanford, where it basically in bacteria will circulate them. But you can only go down to 850 base pairs with that because the DNA is too rigid. Another format that we use is linear DNA. And so we use an enzyme called prototelomerase, which is found in some bugs, and it will actually bind the sequence and ligate the 5' to the 3' and create what's called doggy bone DNA. And so you can create these double-stranded DNAs that are linear, and we can make different sizes of these. And there's a company out there that uses this technology. I think it's called Touchlight. And so you can make little mini DNAs this way. You can see that we can make a 200 base pair. So now we're even getting much smaller. So the question we had was, how small can we go and still have transcription from these DNAs? Because the idea being that if we make them too small, it may be that the transcription factors won't be able to bind and transcribe. If you think about it, everyone's been to one of those like weird circuses where they put the motorcyclists in those metal balls, right? And they like, it's like the orb of death. You can imagine if the orb got small enough, the motorcycle won't be able to go in there. So I think of that the same way when I think about plasmids. If you make it too small, the transcription factors won't be able to move around. If you find yourself at one of those circuses, I don't know, I have two and I don't know how I got there, but so you can see what we did here is we made several different formats. We made the circular, we called these linear ones closed-end DNA threads, mostly to get away from trademarks. And we increased the size, and we increased with size, we can put more copies of tRNAs in. We found that the 200 base pair didn't support suppression, but when we increased the size, that we obviously got transcription and suppression of the stop codon. With the linear, we never got to the same level with the big circles, but at 200 base pairs, we actually got pretty robust transcription of our tRNA and suppression of the stop codon. And so size to us was what mattered. And so we moved forward with the 200 base pair linear thread. And so now we're taking up with our transgene taking up 70% of the space and material. We then transfected these 200 base pair threads into cells that are not dividing. And so the trick that we use is a phytocolon. So phytocolon will stop cell splitting, and you accumulate the G1S phase. And this is important because the nuclear membrane doesn't break down, right? And so if you get function of your tRNA, it means that it wasn't because it got through the broken down nuclear membrane, but in fact, it actually moved through actively or passively to get there. And so we had some cells that had a PTC reporter, so a nanoluciferase, and then also had an M-scarlet, but we'll only be talking about the generation of the nanoluciferase. And when we increase the size of the linear thread, right, you see a size dependence when the cells weren't splitting. So that means that at 200 base pairs, you're able to get these DNAs into the non-dividing cell nucleus, but when you got bigger, size became a barrier. So we thought, can we improve on this? So we thought, can we functionalize our linear picovectors to actually actively move the DNA into the nucleus? And so rather than relying on DNA that we've PCR'd or we've generated from E. coli, we made a completely synthetic DNA vector. And so we order all of the bits from IDT. We have these hairpins at the end that have DBCOs that we can use click chemistry to put functionalities on. So shown on the right, we've actually clicked on a lexafluorophore, and you can see that on the top, we have ethidium bromide, and this is actually the visualization of the DNA in the agarose gel. And on the other side, we've put on different nuclear localization signals. So we put on a peptide that was scrambled. You can see the gel shift. An SV40 nuclear local NLS, Ty1, and an HLA. And then we also put on an alpha peptide, of which there's a nanobody which binds with really high affinity, so we can functionalize it in two ways. So we're just showing you that we can click on a bunch of different peptides, and that now the idea is that since we can effectively get huge amounts of these DNAs that have been functionalized, you can click on all sorts of doodads. And so we're actively looking at ways to increase intracellular mobility, endosomal escape, and cell entry using just one platform. And so when you come up with something like this that you're going to base your lab on and have several papers on, that's kind of a novel approach, you need to have an acronym. Otherwise, you haven't done science correct. And so we were lucky that if you look at all the letters, it's a really roll off the tongue acronym, MOFUS. And so, but yeah, we're going to work on that. Okay, so the other thing we have is this 200 base pair tRNA. And this is the one that we use from the screen. We then optimized with over 600 different nucleotide variants to see if we can increase the expression from this 200 base pair, and you can see that we've done that. So we get 7-fold increase in PTC suppression. So the y-axis is really cool. We're able to make it 7-fold better, but maybe more importantly is the x-axis. So we only need to put in about 150th amount of what we normally would have to get the same PTC suppression. And so when you think about the delivery barrier, this is what's most important. How little can you get into a single cell to give you enough suppression to be therapeutically meaningful? All right, so now we've put on NLSs, we've optimized them, and now we're going to go back into cells that are not dividing to see how much PTC suppression we get. So what you can see in our optimized 200 base per picovector with no NLS present, you get what I showed you before, right? With non-dividing and dividing. But now you can actually get much more PTC suppression with Ti-1 and HLA NLSs when they're appended to it. So this is active transport of DNA into the nucleus. I think what's really fascinating about this is that the HLA NLSs, HLA is a protein that's really important for tRNA post-transcriptional modification. It actually is present for clipping off the 3' end. And so it's interesting to me that the protein that has an NLS that's important for cutting up tRNAs is the one that works the best for getting our DNA into the part of the nucleus where it can be transcribed. And so we have ideas that it may be that getting it into the nucleus is one layer to all of this, but then also getting it to the right part of the nucleus once it's there, where transcription of tRNAs exists, is the other part. And so the one of the issues with using DNA that I haven't talked about yet is its general toxicity. So when you put plasmid DNA into cells and it doesn't get into the nucleus, it activates this C-gas sting pathway. And usually in your labs, when you're doing transfections, you've already scraped your cells and you don't really care, right? But when you're making a therapeutic, this is super important. And so we wanted to know that by putting in our DNA, by actively directing it to the nucleus, can we reduce the resident time in the cytoplasm and reduce the C-gas sting activation? So C-gas sting is just shown here as a schematic on the left. And we transfected cells with a CFTR plasmid, with a plasmid with our leucine suppressor tRNAs, and then a no NLS 200 base pair picovector. You can see that we get a reduction in the C-gas sting, and that's just because the size of the DNA actually matters here. Smaller DNAs don't activate the C-gas sting pathway as well. And when we put our NLS on, you see we reduce this almost down to nothing. And so it has the functionality is important for not only functionalizing the picovectors, not only important for getting into the nucleus, it's for getting it out of the cytoplasm, where you get toxicity, which is equally important. So in conclusion, the endogenous CFTR transcript abundance and channel function can be significantly rescued with our ACE-tRNA approach. And it appears that this is only limited by our delivery efficiency. One ACE-tRNA sequence can rescue functional CFTR encoded from endogenous gene harboring most of the prevalent CF PTCs. And we developed a modular synthetic linear picovector with an amazing acronym for very efficient expression of our ACE-tRNAs. And so future directions are to determine how many CFTR PTCs can be functionally rescued by one ACE-tRNA. And we develop a lot of cloning strategies in the lab. And so using high throughput cloning, we're going to make all 200 nonsense mutations, and we're going to make all 200 leucine variants and all 200 serine variants. And then we're going to test them using high throughput leucine chamber-like experiments to see how many of those nonsense mutations will be susceptible to having a leucine or a serine put in. We're collaborating with Paul McRae and Bev Davison at CHOP. We're still hanging on to some of this AAV. Bev Davison has made really good capsids through evolution in non-human primates. And so we're collaborating with them to still hang on to this AAV idea. And we're collaborating with Alexandra Piotrowski-Daspit at University of Michigan that has really nice lipid nanoparticle technologies for delivery of these DNA picovectors. And we've had some luck already. It's just not CFTR, but we've used ACE-tRNAs for leucine and serine to rescue dozens of SIR1 nonsense mutations to get function, and also REP1, which results in choroideremia. And then our next immediate efforts are with dystrophin. And ultimately, what we would like to do is be able to deliver these picovectors as naked DNA, so lipid unassisted, using clicked-on doodads, so that we can just inject the DNA into the blood and it can get into the cells, or of muscle cells, or inhale it and get it into airway cells. And so, really, for understanding how to do this, you need to scale up. And so, we just received some DNA in the lab. And so these are just the 2 hairpins that we ordered from IDT, right? So 10,000 nanomoles of each of these clickable oligos, and each one of them is sitting in a 50 mil Falcon tube. And so we got 15 mils of oligo, which makes up 14% of the total mass of each one of these picovectors. So that's each tube holds 85 milligrams of oligo. I never in my mind, I never thought that we would be ordering Falcon tubes worth of oligos. And this is the best technician in the world, Katie, who also thinks that it's amazing that the powder at the bottom of these 50 mil conicals is an oligo. This is also super expensive science. So these 2 oligos, this was $30,000. So if anyone wants to get into this business, write a bunch of grants. And then again, that's just 2 of the oligos, but we're all in, we're doing it. So I'd like to thank the key players. And so Wuri Ko was the graduate student who did a lot of the leucine variant experiments, and she got her PhD this year and is now working at Vertex. Jeff Gabel, he did a lot of the really nice, and I didn't get to show all of it, but the flow cytometry data. Emily Sorensen just qualified, and so she is holding a really big wrench. And then Joe Porter is a faculty in my lab, and it's up to you to decide if that's a small wrench or a big. He's huge, or is Emily just tiny? Anyway. So Joe is amazing. We call him Chat Joe PT, or the DNA Pope. He's what's making all of this really work. And with that, I'd also like to thank all the other members of my lab, and of course my funding, which I still hope is there in the coming years. Thank you for your attention, and thank you. Yeah.
I thought the idea of the fact that where the PTC is located, that context was really, really cool. And I was just curious if there was any, if you looked at the sequence context around it, and if that, correlates with the, if for one PTC, the context is the same for another that is hard to suppress, for example.
Yeah. So we know that having a C in the +4 position is difficult to suppress. And that was shown by just our little mini screen. For sure, there's lots of data out there where they've used G418 and other aminoglycosides and read-through agents, and they found that there is a sequence dependence, and it's pretty consistent. So one thing that we're doing, we just recruited Carrie Thrasher to the lab. We're screening all 19,000 NTCs with 300 base pairs of context with our suppressor tRNAs, and we're also studying 9,000 disease-causing PTCs and also poison exons. And so we're going to have a lot of data to mine to see how context affects PTC suppression, and see if it holds up, to see if it's true for all PTCs or if there's something special that's happening that we don't understand yet. Yeah.
Fantastic talk.
Thank you.
On that
first
section where you're showing that dose dependency for the PTC suppression, I was wondering if you have looked at the NTC run through issue.
That's a great idea, because we're doing it. And that's a great question. So
is it ever obvious, I guess? Is there ever cytotoxicity at a high dose?
Yeah, for sure there is. Yeah. You can, it's just like you can have too much water. I like to use that analogy because you need water to live, but too much. Yeah. So for sure, what we think is the therapeutic window is how much read through you get of NTCs, and then backing it off enough that you'll still get enough PTC read through that you don't get NTC read through. So that difference that you'll get in the dose response, we think is your therapeutic window.
And it looks wide. Sorry.
No, go ahead.
I was just saying, it looks wide, like how big.
It's going to depend on the PTC. So each PTC is going to have a different read through ability. And so for sure, there's going to be PTCs in some genes that are like really difficult to suppress. And that really is going to narrow the window. But there's lots of other factors of like how much of a protein do you need to be therapeutically meaningful? So we know for some enzymes, you only need 1% of wild type, right? And so maybe if the moons align, if it's a difficult PTC to suppress, but you don't need a lot of the protein, that it would be viable therapeutically. But for sure, you're right. That window is going to change. And it's going to change not only by PTC, but it's going to change by what tissue you're delivering it to, because different transcripts are expressed in different tissues, and different transcripts are going to have different NTC that are going to be easily or less easily suppressed. And so our goal is to go in with several serotypes of AAVs, get into every tissue, perform ribosomal profiling, and really document which NTCs are easier to read through and which ones are not. Yeah, it's a lot, but you're right. I mean, these are the in question, and these are experiments that companies won't do because they never want to know which NTCs are read through. They just want to have enough to be therapeutic. And so even though we're, I guess thought of as pioneers in this field or whatever, I want to be the one that kills the idea if it's going to be a problem, and why is my approach. Yeah, thank you.
Right at the beginning, John, you sort of blow through the charging problem.
Yeah.
But there's type 1 and type 2 tRNAs, and I can't remember which one, but one of them does pay attention to the anticodon, right? So is some of the differences between the different examples?
100%. Yeah. So you're right. The tryptophan is the one where the anticodon, and this is a cool example of it, even though it's a pain in the ass because it doesn't work well. Okay. So tryptophan, when you make a UGA suppressor, is not good. But when you make it a UAG suppressor, it's one of the better ones. And that's because that synthetase recognizes the anticodon, and that's one of the factors, right? But when you look at leucine, it doesn't use the anticodon. And then when you look at serine, it doesn't. And it
Type 1, type 2, that's a difference?
Yeah.
Oh.
And really what it comes down to is leucine and serine have many more codons that they suppress. And so it has to be sloppy, right? And so it uses a synthetase that, however, chicken or the egg. But because it suppresses so many different codons, it has to be lax in that area or it doesn't recognize it at all.
Maybe I missed it, your readout for some of the data that you show is the current.
Yeah.
If they're current. But we know that like if you mutate a channel, right? You may get bigger currents, but that may also have an impact on tissue function.
Yes. So you get like a gain of function that's inappropriate.
Yeah.
Yeah.
And have you looked into that yet? I mean, like that part of the translational to the tissue level?
We haven't yet. Yeah. So what you could effectively do is not make it responsive to like PKA, or you can make it not responsive to ATP and just kind of be always on. It's possible, right? We don't know. It's a possibility. Like, I guess what would be really, so when we make these variants, it's like, it'll be a very large structure function experiment. And we expect actually that some leucine variants will have gain of function. So actually be super active, better than wild type. And we hope that happens because there are many groups that are actually going out and doing viral delivery of cDNA. And our hopes is that we can actually not make a wild type CFTR, but put in missense mutations that are gain of function that allows you not to have to deliver as much of that AAV and get rescue. And so that's one of the end results of that screen as well. But yeah, your question about, for sure, we're always worried about, are we going to screw things up? I guess we'll find out. Yeah.
Yes. Yep. Okay, thank you, John.
Thank you, everybody.